somewhat different. She's definitely, I mean, so Joseph mentored my oldest son and two of my daughters over at AgroLife, and like my son is very like computer oriented, and if you can get an excuse to use a computer, he'll just do that. My oldest daughter is very much a black and white personality, very outspoken, and then my second daughter is more of the artistic personality where it's kind of free form. She'll kind of do what she wants to do. And
it's funny to watch them all when we go hoe weeds or put stakes out, especially hoeing weeds, because Erin will walk her block and she will not deviate from the rows that she's doing.
Yeah.
Julia maybe 4 studies over
Yeah.
and then come back. And then Brandon gets it done as quickly as humanly possible.
Yeah, exactly. One thing I will warn you about, because I've learned this the hard way on here, is always make sure that it's muted on here. Otherwise, it'll start talking over the top of you about 10 seconds after you're talking. All right.
And is it okay if I step away from the lectern?
Yes.
I have a clicker.
Yeah. Did we get him set up with the microphone for this computer? We've got a wireless microphone. Like I said, Goobon came prepared. Escape.
Okay.
You have to all right, audio settings. I don't think we'll have anyone calling in. All right, Joseph, walk over there and say a few words. Yeah, let's try it one more time. Just say test about 4 times. Oh, it's not there yet. All right, let's try it.
How about now? Is it working now? Do you think it's testing well?
All right, welcome everyone. We're excited today to welcome soon-to-be Dr. Burke to talk to us about soils, to talk to us about soil conservation, and he's got some really interesting things. Interestingly enough, how many of you in here are in the Ag Ed or Ag Com department? Told you there are a lot.
Nice.
Joseph started out here at Texas Tech in Ag Ed and Ag Com, and then he had the wonderful opportunity to student teach at Friendship High School, which is where my children go to school. And he said that the experience was so profound that he decided to go to graduate school in soil fertility. And he's also been a mentor to 3 of my oldest children. I discovered a few years ago that Dr. Katie Lewis, who's the soil fertility specialist at Texas A&M AgriLife and has a joint appointment here, had a soft spot for me going and asking, hey, can you give my children a job? And Joseph had the opportunity to actually figure out what to do with those said children. And so he's worked closely with 3 of my kids. He had a pretty big impact on them because one of them actually changed her major from business to plant and soil science. So thank you, Joseph.
Dr. Ritchie probably had a bigger impact on that than I did, but
No, usually I scare my kids away from that. But I've worked very closely with Dr. Lewis's group and with Joseph specifically for about 5 or 6 years now. We actually have some publications together, which means that he wrote them. He was kind enough to put me on as a co-author, but he has a very interesting presentation. He's an excellent communicator, and he has some things that I think are going to be new and interesting to all of you. So with that, we'll turn it over to you, Joseph.
thanks, Dr. Ritchie. Good afternoon, everyone. It's nice to be here. I don't normally get to work with undergraduates, especially underclassmen, undergraduates. So this is a nice change of pace for me. Like Dr. Ritchie said, my name is Joseph Burke. I'm a PhD candidate at Texas A&M University. And before you throw your tortillas, I did both my bachelor's and master's here at Texas Tech. My bachelor's in this department, ag education. And then I student taught at both Friendship and then at James Madison High School in San Antonio. And both of those experiences taught me that working with parents was not what I should do for the rest of my career. So I then came back for graduate school, excuse me, in soil science, and I've been here for the last 6 years. So interestingly enough, agronomic plant science was the first class I took as an undergraduate when I was a freshman at Tech in 2008. So a way long time ago. But what's interesting is my youngest sister is also in this class. She's sitting right over there as one of her first classes. So hopefully we can steer her out of ag ed and get another soil scientist in the family. So anyway, thank you all for having me here today. Y'all have been learning about agronomic plant science, and I'm going to tell you about what I do, which is agronomic soil science. So very similar ideas, but with a soils perspective. Now, to start that off, we're not going to talk about soils at all, because I want to know, why did all of the megafauna die? And this is related, I promise. So any ideas? If you have the notes, don't cheat. But if you don't have the notes, why did the megafauna die? Ideas, questions, thoughts? Oxygen, yeah. So oxygen concentrations were a little bit higher when the megafauna were here. What else might have been the cause? Not enough food for them, yeah. So if you think back to about 200,000 years ago when the megafauna ruled, the temperature was about 6 degrees Celsius cooler than it was now. So very different plant life than what we have now. What else could have caused the megafauna to die? Maybe around 150,000 years ago. You're all sitting in here today. You're part of the problem. People. So we'll get to climate change. That is part of the problem. But humans are probably the reason why most megafauna died. So if you look at this map around the world, these are the major continents and the major megafauna that were in those areas. So when I say megafauna, I mean huge animals. So think elephants, but everything is an elephant size, okay? So if you ever make it out to the Lubbock Landmark Lake area, they actually have giant statues that are not giant. They're regular size for what the animals that they've dug up there. So they had these sloths that weighed 2 tons. They had armadillos that were the size of a VW bug, right? So really cool things right here in Lubbock that you can go look at. Now, why did that? Yes, Dr. Ritchie.
So real quick question from an outsider. How is it that some of these things did manage to survive, like elephants and giraffes and rhinoceri? You know, if you look in Africa, it seems like we still have a few of them. What was different there than in the rest of the world?
So mostly what happened was people left. Like a vast majority of people left, and agriculture developed there first. So what we'll get to in just a second is talking about why did we hunt these things to extinction. So if you look around the globe, lots of megafauna. The coolest continent to look at this is going to be Australia. And there's actually some really interesting papers and books on why Australia is such an interesting case. So if we look at human migration, humans migrated out of Africa around 100,000 years ago. And this is one of the leading reasons why they think, because it was also a fertile crescent, lots of food, great climate, that we still have megafauna that reside from Africa, right? Elephants, rhinoceri, giraffes, things like that. Humans migrated out mostly to the Mediterranean, and then finally over to Southeast Asia, finally, and then to Australia around 50,000 years ago. And Australia is such an interesting case because once you get there and the ice melts and you don't have land bridges anymore, unless you develop boats, you can't leave. So imagine you put a whole bunch of people on one small space with a limited number of megafauna. You hunt the megafauna to death. What happens? They go extinct and there's no food, right? And so then the people have less ability to evolve because they have less food to feed their brains. And that's why we hunted the megafauna to extinction, is because we needed calories. Our brains were growing, they were doubling every 10,000 years in size. And so by that rate, we needed all of these calories to get that. We couldn't just get it from nuts and berries, so we started hunting these big animals. One thing that humans had this advantage on over other animals is that we can run fast and for long distances. We can't run cheetah fast, but we can run for a long time and outrun the cheetah or any other big animal. So we literally just run them to exhaustion, kill them, and then eat them. And so that's why the megafauna died, or at least that's the leading theory why megafauna died. I will preface this, in 50 years, if we have new evidence, you can't come back and say that I was wrong. I was just wrong at the time. All right. And so again, why do we need those calories? Because our brains were big and we needed more food for our brains. And so the megafauna are the calories that we needed. Lots of fat, lots of protein, perfect things for us to consume in the world. But then around 30,000 years ago or so, most of the megafauna were completely gone. And so we needed to find some other system. We couldn't hunt the buffalo as much as we used to. The buffalo are big, but they're not as big as their woolly mammoth contemporaries. So we had to find something else that we can do. And what's interesting to note is out of that Fertile Crescent, agriculture was born. So the first place that the megafauna left and human beings came out of was also the first place that we developed agriculture. Some archaeologists suggest that it's possible that if humans had developed here in North America, would have been where the Fertile Crescent was, instead of like in Mexico, Central America, was because they just needed to find those food sources first. There's plenty of good agricultural crops that are from this area, but we domesticated other crops first, as y'all have learned in this class. So what's interesting to note, though, is by 1000 BCE, so about 1,000 years before Christ was born, humans had not spread to the Australian continent yet. And so that's why we still believe that the Aborigines were a slowly developing civilization, is because they didn't have lots of calories for their societies to expand like other societies did. So what does any of this have to do with agronomic soil science? I like to claim that soil scientists are the reason why we have civilizations today, because not only did we domesticate plants, we domesticated soils. And that's what we're going to talk about today, is how do we domesticate soils and use soils to grow plants so that way we can sustain human life. Okay. All right, now the megafauna dying off was a big problem, and we came up with a big solution. It was called agriculture. But in modern days, we have big problems still, and these are some of the big problems that face soil science. So we've got wind erosion, climate change, which y'all brought up earlier, water erosion, aquifer decline, which is really big in this area. We're going to talk quite a bit about that today. Desertification, eutrophication. When we apply too many nutrients, they pollute the waters and cause a flush of algae. So salinization from bad water sources, and then mine reclamation. These are all the things that every soil scientist I know deals with, even on a daily basis. We all try to figure out how to solve these big problems. And so if you like solving big problems, soil science is the field for you. Also, it pays more than animal science. So yeah. All right. So agronomic soil science. This is broadly the study of soil systems for the primary purpose of producing food, fiber, feed, and fuel. So everything that you're learning about agronomy, we do that, but with soil science. And the things that we're most interested in are three categories: physical, chemical, and biological properties of soil. So within physical, we talk about texture, aggregate stability, soil structure, the bulk density. These are all terms in soil science that you'll get to know. We're not going to go over them today, but just know that they're important. In the chemical area, the biggest one that we're most worried about is pH, because pH is the master variable in soil chemistry. It controls everything. If you have too low of a pH, you won't have certain things available. Have too high of a pH, you won't have other things available. So we have that sweet spot, and almost all crops evolved within that sweet spot. There's a lot of work in breeding to try to make plants move outside of that area, but really we're kind of concentrated on those thoughts. And then biological, and this is the big one. So if you're going to go into agronomy, if you're going to go into ag ed, and so of the half of you that are ag ed majors in here, most of you will end up becoming agronomists of some kind, because you find that you make way too little money for all the hours that you work, and you can make a lot more money selling things to people than you can teaching kids about the future. So when you come over, remember that you will be selling biology to people. That is the big push right now in the world, is how to sell biology. We're nowhere near figuring it out, but maybe by the end of my career, we will have some idea of how to harness biology for our purposes. Which of these factors do you think is the most important factor overall? Sorry, which one of these individual factors is the most important thing in soil science? It's a thing that every soil scientist studies, whether they like it or not. Biology, but there's one key one within biology. Yes, soil organic matter. And that shows up really poorly on the screen. I should have field tested this before I came in here. But soil organic matter is the most important thing that every soil scientist studies. And the reason why is because it controls every single one of these other factors. Every single one of these is touched by soil organic matter, and primarily soil organic carbon, which is the biggest component of soil organic matter. My specialty area is soil organic carbon, along with like half the other soil scientists in the world. So I just have to talk better than they do so I can get jobs compared to them. All right, so what is soil organic matter? Anybody want to take a gander? Farm kids usually always get this wrong. What is soil organic matter? That's a good one. What else? So I'm going to challenge you on that one though, because is it fresh residue or is it partially decomposed residue? Exactly. So plant litter will be partially transformed by insects and microbes, will be cut up into little pieces, and they start this decomposition process. And once they produce biteric acid, which is one of the key steps in a microbial decomposition, it becomes soil organic matter, okay? And that's what we work with. Plant residues are great, don't get me wrong, but what we want to get to is soil organic matter. Within most soils, we're talking about, so this is all of soil here, between 0.5% and 6% will be soil organic matter. So a very small fraction is soil organic matter. Within that fraction, we have this stable fraction, something that's living. So that's microbes that are actually living, doing these processes. And then there's going to be active soil organic matter. And that's stuff that reacts with clay minerals, provides nutrients, stores water, things like that. So a very small portion of our soil is going to be organic matter, but it plays the biggest role in everything else that we do. What do you think are the average soil organic matter levels in West Texas? Let's say Lubbock County. Average soil organic matter. Throw a number at me. Between 0.5 and 6. 1 is a very good guess. Usually double what we see though. So we're hovering around 0.5% organic matter. In my study in Lamesa, it's been going on since 1998. I was 8 years old then, so I was not involved, but someone started the study in 1998. And from 1998 till now, we have doubled our soil organic matter from 0.5 to 0.8%. So in 23 years of no tillage, cover crops, the best that we can throw at it, we have doubled our soil organic matter to a very, very small fraction, okay? So it takes a long time to build up this stable fraction of soil organic matter, but it's super important that we study it. All right, so as soil enters, right, so plants die, they exude exudates, their roots are leaky, they leak things out, that enters this active fraction, and it decomposes really quickly. You're gonna lose about 90% of every piece of plant biomass that you add to the soil as CO2 back to the atmosphere, okay? Remember that because that's important for climate change, all right? So that rapidly decomposes. That little teeny tiny percent is going to go into this slow soil organic matter fraction. So that's going to slowly turn over. And this takes eons for us to build this fraction up. And then a teeny tiny percent of that is going to enter this passive pool. And this stuff is really the stuff that sticks around forever. That's why that number is from 0.5% to 6. It always hangs around 0.5% because we have this stable pool that just keeps everything there and doesn't like to decompose. It doesn't like to react. It just kind of hangs around, right? And so what we want to do is we want to slowly build this pool because this is what makes our clay particles, which is the most important part of our soil texture, to stick together, cause them to flocculate. right? And so once they flocculate, we can build everything else. We can build aggregates, we can store water, we can provide minerals. And so we want to get to that active pool, but it takes a long time and we lose a lot of CO2. Yes, sir.
So how deep is this like passive like layer? How deep it is in the ground?
It depends on how much water you get. So passive soil organic matter is so small that typically it will translocate within the soil profile. So it'll move with water into the soil profile. But typically most of our organic matter is going to be in the top 2 to 3 inches of soil. So that's why your soil has a darker color on top, because organic matter is going to have a brownish tinge. And so that's why soils are darker at the top than they are at the bottom. Yes, sir.
So back when I took my intro ag class, we always talked about how you can't get nitrogen out of the system if your carbon to nitrogen ratio is too high. But for the passive soil organic matter, we have a relatively small carbon to nitrogen ratio. So what happens in that case? Is that nitrogen still just kind of bound up and not available, or what's the situation there?
Yep, great question. So why isn't this decomposing faster, right? The lower you get on the C to N ratio, the closer to 1, the faster things break down. That's why when human bodies decompose, their skin sloughs off and like they turn to liquid, but their bones stick around. It's because our internal organs have a really low C to N ratio, whereas our bones have a really high C to N ratio. So if you want to know about decomposition, it's all the same for human bodies or not. But what happens is there's a small fraction of that passive fraction, that it's structurally bound. And so you have this lattice network of things called lignins, and they just hold things together so they don't decompose very quickly. So you may have an average C to N ratio of 7, but it's bound inside all of these little things inside of it. Good question. So not all soil organic matter is treated the same. So what are the benefits or the functions of soil organic matter? This is kind of what I was alluding to. So biological function, it can provide energy for microbes, right? Microbes are doing all of this nutrient cycling for us. It can provide actual nutrients itself, nitrogen, phosphorus, and sulfur. And then it also contributes to the resilience of the system. So biology, let's say we have a bad weather event, it comes in and kills everything off. If you have more biology through soil organic matter, it can help protect those communities to respond and come back faster. Chemical functions, it contributes to cation exchange capacity. That's how nutrients are exchanged. So if I need a nitrogen, I have to give up a hydrogen and it takes it up, that kind of thing, it provides those to it. It can enhance pH buffering. This is really big if we add a bunch of fertilizer, because nitrogen-based or ammonium-based fertilizers, they produce a lot of acidity in soil. And so this helps buffer against that acidity. And then it complexes cations. So things like ammonium and magnesium, it can bind those together and hold them in place, specifically iron. And then physical functions, so it improves the stability of soils. It just makes them more resilient. It can influence the water retention. So we say that soil organic matter sucks, and it just holds a bunch of water in place that you can then use later. And then finally, it alters the thermal properties of soil. So you have a whole bunch of this dead mass on something, it'll help protect it from the sun's effects. And so we'll see that a lot in our soil systems. All right, now why do we care, right? It just sounds like a whole bunch of mumbo jumbo, doesn't really apply to us in real life. Well, the Dust Bowl is the perfect reason why it does apply to us. So Dust Bowl, if any of you are from this region, you can still see the scars on the earth is what old people talk about. And you can still see where the soil used to be at one height and they had a T-post, or not a T-post back then, but maybe a wooden stake or a wooden fence post. And now their soil is here. And so their fence post is exposed an extra couple of feet. And that's because of the Dust Bowl. But we still have Dust Bowl events literally every day, right? So this was from when I was not undergrad, I was visiting Lubbock. This is Terry County, just southeast of here or southwest of here in 2013, one of the worst ones I've ever seen. And actually funny, I was an undergrad in 2011 when we had one of the worst haboobs ever in Lubbock. And I was walking from my chemistry class back to my dorm. I was a CA in Stengel-Murdo. And we had to wear our chemistry goggles to be able to see outside. The street lamps came on at noon and you couldn't see maybe 3 feet in front of you. There was so much soil in the air. It was insane. So anyway, bad dust storms. I didn't have an iPhone back then though, because they were brand new. To date myself at all. This is a picture from 2019. This is our research center looking west. It's kind of hard to see in this picture, but it was about 20,000 foot tall cloud of dust that was coming for us. And this was me driving home earlier this year in January. just trying to get home in a dust storm. So it just goes to show you how bad the dust storms can still be. But if you think about it, since the Dust Bowl, we have reduced wind erosion in this county by 80%. So we have taken this to this and cut down the number of times by 8 in a 10-year period, right? So this used to happen every year. Now this happens every couple of years. And if you look at this year, the last 2 years in a row. So it's really important that we do this work because we can conserve our soils and keep them in place. When soils blow, because we have our organic matter at the top, you also lose your organic matter. And so every time that soil is blowing away, you're losing a little bit more productivity of that soil. Now, the nice thing is if the soil blows onto your land, you're getting a little bit more productivity every single time. So that's why we encourage cover crops, because you can grab some of your neighbor's earth and hold it in place for you. Or as our vineyard people call it, an uncalibrated sprayer event. That's what they call dust storms. It'll be funny when you take some upper level classes. All right. All right, so this is what we're talking about. So this is how we prevent wind erosion on our research farms here. So here's our cotton crops here. This is going to be a rye or wheat cover crop that we planted in between the beds of these. And what it does is it, one, doesn't allow the particles to start moving, because as the wind blows, those particles start to hit each other, and they move faster and faster, and they hit more and more particles, and they get swept up into the air. This slows that down, right? It's much harder to run through a crowd of people than it is to run by yourself. That's why we run track in our own lanes, right? So, and then what it also does is if you do have sand or clay blowing from your neighbor, it'll hit this and it slows it down enough that where it settles, and then it doesn't move. So then you are picking up soil. I'll have a picture here in a little bit. We've measured on our cotton plants after windstorms in this study, and we can gain up to 1 inch in a single windstorm of soil. Just because of other people's next to us blowing away. All right, so why don't we plant cover crops everywhere, right? Why don't you just drive down the road in December and you just see cover crops, beautiful wheat crops everywhere? Well, the reason for that is twofold. One, they take up water, and we have limited amount of rainfall here. And two, they tie up nitrogen, because as they decompose, that nitrogen gets bound inside of them, and microbes want to use that nitrogen for something else. So this is from 2017. This shows the average percent of cover crops grown in a total cropping area. And the maximum value that you can see down there, it's easier on your slides, is 25%. So in the entire region, the maximum it got to was 25%. And I summarized these. The highest it actually got to was like 24.5. So I summarized it. The greatest county for it was right over here. I think it's Borden County. And they had 24.5% of their growers produce cover crops on their acres. So really small amounts. But reduced tillage, one of the things that we'll talk about today, is around 50%. It's real easy to eliminate tillage. It's not so easy to implement cover crops in our systems. And as a researcher, my job is to study why are those numbers so low, and how can we increase the rate of adoption for those things. So one of the things that also makes it challenging is how much time we have to actually plant a cover crop. So we will normally plant cotton in mid to early May, and then this year in July, because we had a tornado go through our research plots. So we're not harvesting stuff in July, because that'll mean we'll harvest in February once we get a freeze. And that's just not going to happen. So anyway, we normally plant our cotton in May, and then we'll harvest it between now and mid-November, hopefully before Thanksgiving. We've harvested all the way up until Christmas, but we had a warm fall that year and we didn't have to. But then I have to plant a cover crop, and most crops don't like Lubbock winters, right? I don't like Lubbock winters. You probably don't like Lubbock winters. It's cold, it's windy, it's nasty. They don't like to grow outside either. And so we have a really narrow time period from when we get that cover crop out of, or the cotton crop out of the ground, to plant a cover crop behind it. And then I need to terminate that early enough to where I can recharge the water here, so that way I have enough water to plant my cotton crop into. Because again, we're a water limited system, about 18.5 inches of rainfall a year here in West Texas. And it takes what, 19 inches of rain to produce a cotton crop? So if on an average year we get 18.5 inches, and I'm going to use 19 inches of that to grow a cotton crop, there's nothing left for my cover, right? So that's one of the biggest concerns, is farmers do the math with rain, and they're not going to take the gamble on, do I have a 2021 where I'm going to have a lot of rain, or am I going to have a 2011 where I get 1 inch of rainfall in my growing season, right? So that's the calculus that we're trying to do. So it makes it difficult in these systems. Luckily, I work in research, which isn't the real world. And so I can design experiments and have other people pay for them. And so the one experiment that I work on, this is in La Mesa. So if you're coming up from the San Angelo area, probably drive through La Mesa every time you come up. I'm off to the side. You just wave at me when you drive by. Our field is right by the highway. And this study was established in 1998. So some of you were probably, well, actually, almost all of you were probably not even born yet, if I had to guess. Was anyone born before 1998? Oh, wow, that hurts. Okay, so when your parents were thinking about you in a few years, then this is when this study was established, okay? And so there's three major systems that we're looking at. We're looking at continuous cotton with a conventional tillage. This is traditional practices, everything that we do all the time. We also looked at no tillage with a rye cover in cotton. So we switched to no-till, we planted a cover crop in the wintertime, but it's still always cotton after cotton. And then we had a mixed species cover. And this is based off NRCS recommendations. These recommendations have now been changed because of this research project, not because of me, but because somebody else wrote them. And so this was the old recommendation. Now, the problem with our legumes here and then our radish is it gets way too cold for them to grow. And so essentially what we had is we had 45 pounds to the acre of rye, and then we had like 27.5 pounds to the acre of small rye. So what I like to say is we had a rye cover crop and then a very expensive rye cover crop because of all the legumes that were in it. And we compared those against each other. And what we wanted to see was not only what was yield differences, what are the nitrogen differences, but how much water was there, because that's the key issue. And that's one of the papers that I didn't want to write in my thesis, and Dr. Ritchie really encouraged me to. It was the last chapter I wrote. It was the most boring chapter, and it's the one that's elevated my career to what it is today. And so it's funny how things turn around on you. But so we're going to start with carbon, because carbon is organic matter, right? Organic matter is 54.3% carbon. And so if we study the carbon, then we can understand everything else that's happening with organic matter. So we have our three systems, our conventional tillage, we have our no-till with a mixed species cover, our no-tillage with a rye cover, and then I compared it to a native rangeland just because I could and I had the money. So what we saw was that with the addition of our rye cover crop or our mixed species cover crop and no-tillage, we significantly increased the amount of carbon in our systems over 23 years. And there was no difference between our systems and a system that had never been plowed ever before, right? Had never seen a plow before. So we were doing the same ecological benefits as a no-till or a native system could. And that's what everybody wants to compare back to, right? They say, oh, if we could just convert everything back to grassland and raise happy cows, all the problems in the world would not exist. That's not true. And we're showing that we can do the same benefit as if you were growing a crop compared to if you didn't have a crop. And then this over here, so this is my pet project. So potassium permanganate oxidizable carbon is what I started my career studying. And it's just this really fancy term for purple carbon. And what we found is the same trend between these two systems. So, but we wrote the very first paper on epoxy in semi-arid systems. Go check it out. It's only gotten like 5 citations, but maybe someday they'll realize how important it is and they'll keep citing our work. So anyway, so water. Okay, let's talk water. So this is starting in 2016 and running through 2018, okay? And this looks really confusing, but I promise it's actually a very simple figure. What we look at is we have our depth below the soil surface. So our soil surface is about right here, and we're looking at 20, 40, 60, 80, and 100 centimeters below that surface, okay? And we're looking at how moisture changes. So the more red something is, the less water that's there. The more blue or purple something is, the more water that's there. And then green's kind of in the middle. What we see comparing our conventional to our rye is that when we terminate our cover crop, we have less moisture available in our no-till systems compared to our conventional, which we expect to see that, right? Our cover crop is actively growing, it's taking up moisture, it's utilizing something that the conventional tillage isn't. So it's going to drain that soil profile. But as soon as we terminate that cover crop, we start to see a significant increase in water compared to our conventional tilled system. So that organic matter that we've added through that biomass, what it's doing is it's holding water in place, keeping the soil from, it's like shading it so it doesn't evaporate as much water off the top, and then providing more water there inside the soil for that cotton crop. And we see that throughout the growing season. What's also fascinating to see is that we have less reductions in water following that cover crop in the middle of the season. So we have two cotton plants growing next to each other, one in conventional, one in no-till rye. And what they're doing is the no-till rye is holding more moisture for that cotton crop compared to the conventional tilled system. And so we have more water later in the season. Does that make sense? Okay. So that's a good thing, right? So what if our irrigation runs out? What if our pump breaks? What if we have a dry spell? We have more water available to our plants in this no-till system compared to that conventionally tilled system. So it's making us more resilient, and that's what we want to be. Resilience is the word of your generation. We want everything to be more resilient. All right. Now, the question that I always get, and I always have to throw this in here because I work with farmers a lot, is, well, what about cotton yields? Do you still make as much money? Not yet, but we're optimizing the systems. Okay, so if you look at 2016 and 2017, those are the only years we've seen differences. With the conventionally tilled system, we always have more lint produced, so more money than we do with our conservation systems. Other years, there is no impact statistically. There is a numerical impact, obviously, but statistically, there is no impact with our addition of treatment. So we're not doing more harm than we normally could. But we need to figure out, why are we seeing that yield reduction? Why is there a little less yield in our no-till rye systems compared to our conventionally tilled systems? And the answer comes back to nitrogen. So if you can't study carbon, the next coolest thing to study is nitrogen in soil science. And so to evaluate that question, because as scientists, we don't just stop and say, here's what happened, we go on to the next thing. We developed two cropping systems, one in Lamesa and one in Halfway, Texas, which I finally learned on Monday was halfway between Olton and Plainview. I never knew what it was, but it's the halfway point when you were traveling from the county seat in Olton to the county seat in Plainview. So fun fact, if you're ever going through there. And so we have two replicated studies. They're the exact same. One is a continuous cotton winter fallow system, so the same one we talked about earlier. The other one is continuous cotton with a cover crop. And then we have a cotton wheat fallow, so that cropping system rotation. So cotton, then you plant wheat as soon as you harvest your cotton, and then you wait a year after you harvest your wheat and you plant cotton back into that, okay? So cotton wheat fallow rotation. And then we have it replicated another way. So wedge 3 is cotton wheat fallow, wedge 4 is wheat cotton fallow, so that way I have data every year for my study, okay? So this is replicated. And with carbon, we see a very different trend. And one of the reasons why we see this trend and we think it's going on is because this is a much younger system. This system was initiated in 2014, and we don't think we've got to that threshold carbon point that we need to get to start to see the benefits in the system. So there's no differences between treatments except at this top soil depth, where we had more carbon with our cover crop system than we did with the other systems. So still working on this, paper soon to be. If you really need something to fall asleep to at night, happy to email it to you. All right, so now for the nitrogen question. We know that there is a deficit we know that there is a yield drag. We know that we have less lint produced in our no-till systems compared to our conventional. So one idea that we had is, because you're adding a bunch of carbon with our biomass, but we're not adding any nitrogen, maybe we just need to up the nitrogen just a little bit to feed the microbes so they break down the nitrogen that's in the biomass, right? So one of the systems that we designed, so this is our continuous cotton winter fallow. So this means no cover crop. And then we have a cover crop. And we added nitrogen at 3 different stages: right before we planted by about 2 weeks, when we planted, emergence with 3 weeks, and then this is called pinhead square. And this is when the cotton plants start to develop their little flowers. So this is the point where they're taking up the most nitrogen. So we thought, what if we just add a little bit more nitrogen right before their highest demand? And what we found is that by applying that nitrogen 2 weeks before planting, we actually gave those microbes enough opportunity to break down that biomass that's there, releasing that extra nitrogen into the system, and then you have greater lint yields. So we figured out the system, right? We can solve everything now. We know we have more water. We know we have more nitrogen. Unfortunately, 2019 was the only year I could get these results to work out. 2020, I thought was going to be fantastic, but we had to replant, so I lost my data. 2021, I was like, this is my last year. I'm defending my dissertation in 2022. I'm going to get it right in 2021. And then we had what's called an inland vortex, which is essentially the amount of wind that's inside of a hurricane is dumped at one time when the air inverses in La Mesa. And it caused essentially an F5 tornado to land right over the top of our research plots. So we lost our entire research station this year. And so I'm just going to graduate with 1 year's worth of data and hope that 2022 is the year that we figure out the system. Okay, so keynote when you go to grad school is you're going to fail a whole bunch and it's going to feel really demoralizing. And you'll have these existential crises where you just feel like you've never been good at anything your entire life. And you're probably right, but you just have to keep going the next day and the next day and the next day, and maybe you'll figure it out. Okay, that's what I keep living for. It's like that gambler's high. Like the next hit is the one that I'm going to win all the money. And that's what I do for grad school. I just do it every day, right? For like $30,000 a year. It's fantastic. All right.
In addition to carbon and nitrogen, do you also consider other nutrients?
Yeah, we look at the whole gambit. So we look at all the macro and micronutrients as well, but my focus is on carbon and nitrogen cycling. All right, so I'll leave you with this. We're going to talk about last two things. Soil health. Has anyone ever heard the term soil health before? I just said it. Anybody? Okay, if you are anything involved with agriculture at all, you will hear the term soil health from here until the end of your days. And I hope that that day is soon, because I don't want to hear the word soil health anymore. It is a trend in soil science. It is for soil fertility 3.0. It used to be soil tilth, then it became soil fertility, and then it became soil quality, and now it's soil health. And essentially, we're just doing the exact same thing, but we add a new buzzword to it, so that way soil scientists can get more money. That's essentially what we're doing over and over and over again. But the definition, the current definition of soil health, according to the NRCS, is the continued capacity of soil to function as a vital living ecosystem that sustains plants, animals, and humans. And it brings back this idea of the chemical, physical, biological, right? When we talk about agronomic soil science, it's just stealing our things over again. And one of the things that soil health does differently than soil quality, which we'll talk about in a second, is it includes both inherent, so what's there and you can't change it, and dynamic, the things that you can change. Okay, that's the main difference between soil health and soil quality. So I made this figure earlier this year. This is just looking at the timeline of soil health. So I went through the literature and found every mention of something to do with soil health, and I tracked it graphically. Okay, so the first mention was in the Dust Bowl in 1931. There was an extension publication by Texas A&M Agricultural Experiment Station looking at what they called soil tilthability in the Texas High Plains. And you know what they recommended to reduce your soil's ability to erode? Cover crops and crop rotations. So literally nothing has changed since 1931. We're doing the exact same research they did all those years ago, just with more advanced tools, right? And then we fast forward, we developed the Soil Conservation Service. We did some work on how does your soil rate to others. We added biology in the 70s because in the 50s, they discovered the double helix. And then we had all these molecular tools and people had computers. That was the main thing that made biology take off, is people had computers to use. And so they didn't have to punch the data by numbers anymore. And so then in the 1980s, this is when things really changed, because we had all of these new modern crop varieties in the 80s, but we didn't have fertility recommendations. So all of our crops were doing poorly because we weren't adding the right amount of nutrients to them. So the 80s, we changed that. Fun fact, we still haven't updated the fertility recommendations since the 80s. So we have the exact same problem that they had in the 80s when they were thinking back to the 50s, but nobody wants to give us $3 billion to study how much nitrogen cotton needs. And that's about how much money we would need to figure out all the nutrient requirements for modern cotton varieties. So then you fast forward, and then really in the 2000s, that's when soil health took off, because the NRCS, the Natural Resources Conservation Service, had this system called soil quality. And they did such a terrible job unveiling it and recommending it to farmers that nobody took it on. So from 1999 to 2001, they used soil quality. In 2002, NRCS said, you know what? Soil quality is old news. That's bad data. We're going to talk about soil health now. And it's literally the exact same thing as soil quality, just with a new name. Okay. And so now we fast forward to the late 2010s. The Soil Health Institute exists, and their job is to study all things soil health. And so they contract out to people like me who go in and do soil health research. So lots of soil health work. In 2015, we had the International Year of Soils. That was a global thing, hence the international part. And they really helped develop the idea of what global societies need to work on for soil. So we obviously have been tracking soil health because that's what pays the bills. And what we have seen is consistently where we have a cover crop compared to our conventional or our native system, we have greater soil health. So if you do the right things that they've been recommending since 1931, you will improve your soil health compared to your conventionally tilled systems. However, it's going to take a hell of a long time to get there. Now, one last note, anybody whose parents farm, you may want to go home and farm. We have looked at how these biological indicators, because that's what everybody's interested in is soil biology, how they relate back to yield. Never once in the 5 years that we've been tracking soil health has any soil health indicator been connected to yield. There's never been a relationship there at all with statistics. So if a salesman comes up to you and says, I have a, and if you just apply seaweed extract at this rate across your field, you're going to double your crop yields. And if your parents sell bugs in a jug, I apologize in advance, you can come yell at me afterwards, but nowhere have we ever seen a relationship between soil biology and yield, never. And if you look globally, no one else is really seeing significant relationships between biology and yield. There's something else going there, and it's probably the chemistry, because that's usually the thing that dictates plant growth the most. All right, so I am a glass half full, half empty, depending on how many cups of coffee I've had kind of guy. And what I can say is that generally, I believe that there are opportunities, not problems, facing us in the world. So all of these things are opportunities that you can build a career off of. You can study as a soil scientist, and we have solutions for, we just need to adapt them to their climate. So if you like working outside, this is the sales pitch for soil science. If you like working outside, or if you like working in the office, if you like working in a lab, those are all places that soil scientists work, okay? And we work on these big societal issues every single day, trying to understand how can we take something in the field and make it work for the people that are our producers. So is that a cotton producer? Is that a landowner? Is that a suburban mom who's tired of her grass looking crappy at the end of the year? That's what soil scientists do. We try to find solutions for problems. And so if you like problem solving, and if you like putting on that cape of soil science and going out and trying to save the world, this is a field for you to go into because there's so many opportunities, so much funding, and fun fact, there's no difference in pay if you have a bachelor's or a master's degree, so you don't have to go to graduate school if you don't want to, and you can still make a difference. So I recommend all of you consider it. May have not done the best sales pitch today, but if you have any questions, I'm happy to take them. We can talk about the data, talk about different practices, concerns, comments, objections.
Let's give him a round of applause. You make more with a PhD?
Yes, PhD is nearly double your income. So if you can put up with it, you can double what you can make as a soil scientist over a bachelor's degree. Questions, concerns? Yes, ma'am. So the most common nitrogen deficiency you're going to see in cotton is just going to be stunted plants, right? So they're just going to be smaller than everything else. If it's really severe, you're going to get this yellow chlorosis color to it because it's not producing enough chlorophyll. But most of the time, you're just going to have really small plants, very little yield. They're just going to look bad compared to plants that have enough nitrogen. Others? So the ones that we, okay, so I will say that we just took a little bit of nitrogen that we apply later in the season and applied it earlier. So we apply nitrogen both pre-plant, at planting, and then around pinhead square. Yeah, so we use fertigation, so we can apply it through the pivot system, but we also apply it in the seed bed at planting. Yep, good question. And this is all liquid fertilizer. I don't recommend spreading dry fertilizer unless you're in a dryland setting. Other questions? No. So great question. So kind of like what Dr. Ritchie was hitting at, when you add organic matter to a system, things decompose at different rates. So when I was at Friendship, I taught forensics. That was the class that I did my student teaching in. And so I somehow always come back to this idea of human bodies decomposing, because we've all watched CSI, we've all watched one of those shows. And so when a human body is here and then a human body is here, if one is drier than the other one, they're going to decompose at different rates. But as they decompose, different parts of them decompose more quickly, right? So your proteins, which are full of nitrogen, decompose more quickly compared to your bones. Same thing with plants. If you think about a leaf of a plant, lots of proteins, a stem of a plant, not very many proteins. So that's why you see lots of stems, not a lot of leaves. That's why when trees fall over in the woods, you see the stalk or the stem laying there a long time, or the trunk, compared to the leaves in the field. Same thing. Yep. So no, not all organic matter is the same. Others?
Yeah, good job. Boy, disposing of a body is a lot more complex than you think it is.
Not giving recommendations on that, by the way, if you have them.
Yeah, that's right. Yeah, I did tell them that I'm a grave digger.
Yes.
What kind of a soil would you want to put a body into so that it decomposes?
One in a wet area. We want lots of water. Other questions? All right, well, it was nice talking to you all. Here's my email. If you have questions, concerns, if you need a job, I'm hiring 2 student assistants right now. So so as my sister said, she doesn't recommend working for me. I'm a bit of a challenge to work for, but once you get up to speed, everything's great. So, right. Well, thank y'all for having me. If you have any questions, let me know. Good, thank you.
Good presentation.
Thank you.
I went there.
So Josh Anderson and
Yeah.
Jerome McMullen.
Okay, gotcha. Yeah, I love that.
Yeah, it's so nice there.
That's funny.
Oh,
Thank you.
no, we didn't today. We will next time. So you just showed up for free today.